does it look correct? Not now. Now there is this session. Yes, looks correct.
Yes.
Great. So let's start this session. As it was already said, we had one session last week, and today we have this session, and the third session will be next week. And that is the plan that we agreed in this webinar series. So first week was about ecosystems. Then this week we are going through this workflow, what we do in the microbiome data science workflow, what each steps there are. And this third week, next week, we are going to focus more on this statistical analysis. So the thing that we actually do in this MIA ecosystem. As a reminder, these were the take-home messages from the last session last week. So one main point was that there are several different ecosystems, also in Bioconductor, but this tree-summarized experiment ecosystem is the one that we are here focusing on these webinars. Then I forgot to mention that some of you asked that about the micro echo, and was it the micro table? I believe it was the name of the class, and some of you asked that if that was a data container, and it is. I checked, so that is very correct. But as I said, we are here focusing more on this tree-summarized experiment because that allows us to also do metabolomics and proteomics and genomics, and because that's a general purpose data container. Great. So let's focus on this today's topic. So that is to understand what different tasks we have to do in this microbiome data science workflow. So first, we have to, of course, take the samples and then do the sequencing, and that is done in wet labs. And the output of this step, we have the sequences from each sample, so we have to map them, and that is called the upstream bioinformatics. There are many different names for that, but if we use also downstream, then it might make sense. So this downstream, on the other hand, it refers to this statistical analysis. So when we have this data table, abundance table, then we do the statistical testing, for instance, analyzing whether there's association between microbial profile and some disease, for instance. And this is the overall view overview of this workflow. So here are several different steps, and if we zoom a little bit, this is the first part of this workflow. So here is the data generation, meaning that we take samples, then we sequence them, and the output from those sequencing machines are the sequences. So FASTQ or FASTA files. Then we have to do the upstream bioinformatics. So here it is say pre-processing. So we have to map those sequences to taxonomy or functional profiles. And third step is that when we have this data, then we import the data to Bioconductor to this TreeSummarized experiment data container. And once we have the data in the data container, we can start this statistical analysis, so doing agglomeration, data transformations, quality control, and these statistical tests including alpha diversity and differential abundance testing, for instance. And ultimately, we get this very nice report that can be then reproduce the results, and we can share them. So, a little bit about the data generation. There is, of course, because microbes they affect the whole world. So there are different study systems that people research. And for instance, there is one can take environmental samples, or then from one can research human gut. So for instance, in this human gut, then we usually take fecal samples. And after we have taken the samples, then as I mentioned in the last week, I'm not too familiar with these procedures in wet lab, but we do. It's magic for me, so they do magic steps that we get the sequences. So this raw data that tells what sequences we have found in those samples. And there are two ways to sequence the data, so 16S and then shotgun sequencing. And this 16S amplicon sequencing, we get the information who there are, so what bacteria there is present. We get that same information from shotgun sequencing, but in addition, that information that who there are, we get the information what they can do, because we get information also the genes that they are present in a sample. But this third step, if you want to know what they are actually doing, then we need additional data, transcriptomics or metabolomics, and this already highlights the importance of having this ecosystem that can handle this multi-table data. So, for instance, in shotgun sequencing, we get two tables, taxonomy and functional profiling. And this 16S sequencing means that we target this 16S region from this DNA, and this is a very cost-effective way to do. And usually those workflows are very and databases are very established. So because that sequencing type has been around many years. But the problem here is that it does not give you the functional annotation. And secondly, usually this is limited. This resolution of the taxonomy information is limited to genus level, meaning that the species level identification is not always reliable. But if we want better resolution, more information, then we do the shotgun sequencing. And that means that we fragment all the DNAs in the data and then randomly sequence them. And this means that we get much better coverage of the overall DNA, because we get also in Amplicon, this 16S sequencing, we got only information on this specific region of DNA. But here we can have a much better coverage from the whole DNA, also the other genes in the sample. And this means that we get the higher resolution, meaning that we can identify bacteria in a strain level, and also we get the functional potential of the bacteria. But this is on the downside of this is that it's a more expensive way to do. So after we have done this data generation, this sequencing, as I mentioned, we get the raw data, and we have to do something with the raw data. Pre-processing or the upstream bioinformatics. And here the input is always the same, despite on the whether you are using 16S or shotgun approach. So the input is FASTQ or FASTA files, and also the output is the same. So you get always the abundance table. But whether you are using 16S or shotgun approach, you have to use specific tools for that. And in 16S, for instance, DADA2 and a shotgun sequencing, for instance, Metaflan. And here you can see that this DADA2 is in Bioconductor, but usually this is, especially the shotgun sequencing is very computational. The mapping is very computational heavy, which means that the R is not, it's not suitable for this kind of computing. It's usually done in command line tools. For instance, this Metaflan or Chime or Kraken. Great. And as input of this or output of this upstream part, we get the abundance table, as I mentioned. Then we get the taxonomy table. Then we get the phylogeny tree. And then also we get the sample metadata, including the clinical or environmental or experimental variables. And if you are using shotgun sequencing, we also can have functional annotations in addition to this taxonomy annotation. And in this point, what we do is that we add this data to data container. And this data container that we are utilizing is this TreeSummarizedExperiment. And we already went this through last time, but it is I believe it's good to go through once more, because this is very important to understand this structure. So here is the essays, which is the abundance table, and this TreeSummarized experiment can have multiple abundance tables. Thus, there is these three different tables here. The columns are samples, and rows are the bacteria. And both these row and columns, they can have metadata. Here, the column metadata is the sample metadata. And it can have these, for instance, clinical variables, whether the patient have a disease and blood pressure, that kind of stuff. And here the row data is the taxonomy table. Then we had this row tree and column tree. This column tree, as I mentioned last week, is not too often used, but this row tree, what this does is that it has the hierarchy between different bacteria species, the phylogeny, and that is very commonly used. So, for instance, in our case, we would put the abundance table here, sample metadata here, then taxonomy table here, and the phylogeny we would put here. And if we have some information on whether who collect the data and so forth, we would put it in this metadata. Great. Then I believe that we could go to the demonstration, so live coding, and here we are using this Gupta et al. Data from this publication. So I will open my RStudio now, and you should see this screen, and if not, please tell me. So here in this directory, we have this different data. So we have this abundance table here, then we have taxonomy table, then we have this sample metadata, then we have phylogeny. And we can click this and preview the data. So this abundance table, we can see that the first column here is bacteria names, and these columns include the sample names. And each cell here is numeric value. So this is the output from this upstream bioinformatics pipelines, Metaflan and Chime and so forth. So first what we do is that we load the MIA package with the library function. Takes some time, it seems. Then for this importing this phylogeny tree, we need ape package. So we do this similar thing with this library command. And what we do first is that we have to import these data tables and the phylogeny into the R. So let's do that. Let's put this abundance table to abundance underscore table variable, and we read that with read CSV, the base R function. It requires the name of the file, which is the abundance underscore table and CSV, because that's a CSV file. And then as we saw, the names, the row names, the bacteria names are in first column. So we can specify that with row dot names argument and then 1. And if we check that table, just print, I will print first 5 rows, so we can see that we managed to import the data to R, this abundance table. Everything seems correct. And with the same procedure, we do for the taxonomy table and then sample metadata. So let's do that. First the taxonomy table, then read.csv, and then taxonomy table file, and then the same goes here. We can check that, but I have already done that, so the row names are in the first column, and that is the taxonomy table. Then the sample metadata with the same exact command. Read.csv. Great. Now we have imported, we can see from this environment tab here, that here is the abundance table, then sample metadata and taxonomy table. So we have correctly imported these tables into the R. And then we had also this phylogeny tree. So let's put it to tree variable. And here the function is from ape package, the read.tree. And it requires only the name of the file, which is phylogeny.tree. So now we have all these components loaded into R. And the second task here is to create the TreeSummarizedExperiment object. Good to note here is that the abundance table must be a matrix. So we can do that first with this, let's put it back into this abundance underscore table variable, and we can convert this abundance table. It is currently a data frame, so we can convert that to matrix with as matrix command. And now we can proceed with this, creating this TreeSummarizedExperiment object. So how to do that? There is this TreeSummarizedExperiment constructor function. And here what it wants is the abundance table to essays, the same that we saw in the figure. We name this abundance table to counts, as we see that they are counts, and it was the abundance table. So now we can populate the essays slot. They are called slots. So we can populate that with this abundance table list. Then we have the row data, which was the slot for taxonomy table. And the taxonomy table was in this taxonomy underscore table variable. Then we had the sample metadata, and that is we have to put it to call data, column metadata slot, and that was in sample underscore metadata variable. And finally, we had the phylogeny tree, which goes to the row tree slot, and it was just 3. So now if we press enter, it should create us the TreeSummarizedExperiment object. And it shows that it creates that. And now it's not saved anywhere. So we can save that save that. It seems that I have something wrong with my console, but I have here the same exact code. So we do the same exact command, but add it to TSC variable. And why TSC? Because then we remember that that was the TreeSummarized experiment object. And if we print that, we see that it is TreeSummarized experiment object. And I have said many times that it includes rows and it includes columns, and that is the case. So here we can see from dim that it includes 308 rows and 60 columns. And these rows were bacteria and columns were the samples. And these different data types that we put into this TreeSummarized experiment object, it's good to remember that this is not any kind of black box. We can, of course, fetch them there. We can look them and we can modify them. But this abundance table, for instance, we can see that it's here in the essay slots, in this counts, named counts, and how we can fetch that. It's the essay command, and then we specify the name of the abundance table. That's counts. So that's how we take the abundance table out from the TreeSummarized experiment object. Then we can check the taxonomy table, and taxonomy table was in this raw data slot, and we can use this raw data function, and we can see this table here, which shows the taxonomy for each species in this data set. And then if we look the sample metadata from call data, we can see that this is some data set from fecal samples and I guess from India, and then there is some people that have this colorectal cancer, and I guess there is some patients also that are healthy ones. So this is some clinical data set. Great. And this is good point to illustrate why we use this data container. So as I mentioned, this is there is rows and columns, so we can subset the data, and we can add the subset the data to this TSC underscore sub variable, and this works exactly the same as the normal or traditional or standard rectangular data objects in R, such as matrix or data frame. So we have first the rows, and we can, for instance, take rows 1 and 2, and then we have columns, and I select, for instance, the 10th column. And then we can print the result. So we have now 2 rows here and 1 sample. And if we check the is the abundance table. From this subsetted data, we can see that it has now only one sample and two rows. If we look the row data, it includes two rows, which equals to this found from this abundance table. You can see there's E. coli, and here is E. coli, and here is Alice types, Putrescinis, and here is the same. And then finally we can check the column metadata, and it includes only one sample here, which is the same as found from abundance table. So this is why we use these data containers, because they do the bookkeeping for us. So it automatically selects the correct samples from these different data types, sample metadata, abundance table, and taxonomy table. Then as we are using this data container, the point of having this data container was that then the developers can develop methods for this that are utilizing this data container. So for instance, in MIA, we can found this agglomeration function. And what agglomeration or aggregating does is that it summarizes these different taxonomy levels, so that, for instance, for phyla, we get only what phyla is present, and then summarize the bacteria that is belongs to that specific phyla. And if we can apply this agglomeration and save the resulting object to this same TSC variable. And how we do this agglomeration is that we use this agglomerate by ranks function. There is also agglomerate by rank function, but then it agglomerates the data to only specific taxonomy level that we have to select. But here, this agglomerate by ranks does the agglomeration for all taxonomy levels that are present in this dataset at one go. So all these, if here is the row data, so all these different superkingdom, phylum, class, order, family, genus, and so forth, all these at one go. And if we print the resulting variable, we see that we might think that nothing really happened. The dimensions are still 308 and 60. So what actually happened? These agglomerate data are actually here in this alternative experiment slot. So they are added into this same data object. And that is also one benefit of this tree-summarized experiment object, because then we can have all the different versions of our data into this same data container. We can select this agglomerate data with this alt-exp function. What it wants is the tree-summarized experiment object. It was in TSC variable, and then the name that we want from here. So for instance, phylum. And what it did was that it printed this tree-summarized experiment object. But now you can see that it includes only 11 rows, 11 different bacteria. But still it has 60 samples. So this is now agglomerated phylum level. And this is all these different agglomerated data. They are added to this alternative experiment mechanism. So this agglomerated data is kind of like a tree-summarized experiment inside tree-summarized experiment. And if we print the raw data from this agglomerated data, we do it by this raw data function. But here it's good to note that we have to select the agglomerated data here. So it was Alt-exp. That's how we select the agglomerated data from this tree-summarized experiment object. And then the variable, and then phylum, because we want the phylum level. And we can see that in this aggregated data, we have 11 different bacteria. And all these are now NAs, these lower level ranks. And this is something to expect because we are missing the information when we are summarizing the data to this phylum level. So that is agglomeration, and that is done, for instance, well, as I mentioned, the 16S data, it's the resolution goes up to the genus level. So that might be one reason why we want to aggregate the data to genus level. And then it might be that based on our research question, we are only interested on, let's say, phylum. So that might be one reason also why we want to aggregate the data. Then we can do the data transformation. So in microbiome research, usually we apply different transformations because we have to take into account the nature of microbiome data, the specific nature of that. So for instance, it is compositional, and meaning that these counts itself, they don't really tell much. We have to use ratios instead. So compare, for instance, bacteria A with the bacteria B to in statistical testing. And there are different transformations available, and from online book you can find more information on those. But for sake of example, we can do the relative transformation. And in MIA package, there is this transform essay function, and that includes the standard microbiome transformations that we usually apply to microbiome data. For instance, there is this relative transformation. So what this function needs to apply, that is the tree-summarized experiment object. Then it wants the essay where we want to apply the transformation. So for instance, we want to apply that to counts table. Then we need the method, and it is rel abundance, which is the relative abundance transformation. And what it does is that it adds this this transformed data to the same data container. So if we print that, we see that here are two essays. One is counts, which was the original table, and then we have rel abundance, which is the relative abundance. And we can look the table with this same command as we used for counts. So it was the essay, then the variable, and then the name of the essay, which is rel abundance. And here we can see that these values are not counts anymore. They are from 0 to 1. So they are relative values. Great. And then as a last thing today, we can do some visualization. We can do in the next session we can do much more than this. But today, as a last step, we can do the bar plot, which is very usual way to visualize and summarize the microbiome data. So for that, there is this MiaWiz package, and I load this. And here from loading messages, you can already see that there is ggplot. So everything that comes out from this MiaWiz functions are ggplot objects. And why I'm telling this is that these ggplot objects you can quite easily modify them afterwards. And this is the kind of the standard way to visualize data in R. So in this MiaWiz, there is this plot abundance function. And if we add here the TreeSummarizedExperiment object, and then let's say we want to visualize the phylum level data, and then usually these bar plots are used for visualizing and summarizing the relative data. So relative transform data. So we add here s.relative is true. And it generates us this bar plot that shows the samples in rows. And here in the y-axis there is this relative abundance. And here the colors are different phyla. But this is usually if we are, for instance, interested on different study groups, we want to visualize this same bar plot and show also how they are distributed the samples. So we can add this diagnosis information here in the bar plot. So we can use this same command, plot abundance, then TSE, then rank was phylum, and as relative. And now we can select the call with call.var argument variable from call data. So there was, if I remember correctly, there was this disease was the one of the columns in the call data. Then we want to facet. There is this faceting in ggplots. So that means that we split the plot here into two because we have healthy patients and then the ones with colorectal cancer. So we add true, and then we use the scale because in faceting the scales of the axis we want to select that they are free. Well, I can show you how it looks like if we are not using the scales. So it looks like this. So that is the why we use the scales. You can see these white spaces here. So here, if I go back, so this is the exact same command, and add this free X. It generates this bar plot where we have split these different study groups. And this is very coarse, or how you say it in English, way to do, but this already shows us some information from these study groups. So here there seems to be much more proteobacteria in colorectal cancer patients compared to the healthy ones. And yes, so we can continue from this. We can continue with the alpha diversity and beta diversity and other things in the next week. But this is already really revealing us that there might be some differences with this in colorectal cancer patients in the microbiome compared to the healthy ones. Great, that was my presentation today, and I guess we have still time for questions. Do you want to, Laura, lead this?
Yeah, sure. Thank you so much for that demonstration on how to import data and how to do preliminary analysis, such as creating the graph. We do have a couple of questions. So from the chat, Cynthia is asking, how do they get the data to practice week if they want to follow through?
Yes, there are, and all this, for instance, in MIA package, there is this example data. So was it like this? Yes. So with this data function, you specify the name of the package. All these Bioconductor packages, they have example datasets. And here in this MIA, you can see that there are several different datasets available in this TreeSummarizedExperiment format. And also there are some, if you are referring the abundance tables itself and not the data in this TreeSummarizedExperiment format, there are some available in the online book to practice.
Right. I hope that answers your question, Cynthia. We also have some questions from the grammar pad. Someone asked, should I get really good at data.table package to be able to use Mia package effectively?
Um, so do they need to learn data.table? Was it that correctly? Uh, um, so the data.table is one additional format in R, and you don't need to know that. You don't. You really what you need to know is the basics of R, this data dot frame and matrix. That's all, and rest of these things you just put them into this tree-summarized experiment object, and you can then work with this tree-summarized experiment format instead. So you don't need to know data.table.
Hope that answers your question. Another question is, how can we use tidy models with a tree summarized experiment object?
Uh, Currently, I just guess, not sure. There might be. I can check it for the next session. I don't want to just guess. I There might not be currently support for that. But of course, you can always take the data out from this tree-summarized experiment and then work with this tidy models. But not sure if there is already a support. I have to check that. I will come back to this in the next session.
Okay, thank you. For anyone who's asking about the code, we will share Tuomas's slides that have that code embedded within those slides. We also have documented some of the code that he's been demonstrating on this session from line 65 of the Framer pad, if you'd like to go through now that he's showed you how to get the data. Okay, another question for you, Tuomas. What transformations do you advise for this compositional data?
Well, it's related. Depends on the research question and statistical test that you are doing. So it's there are more and more papers on benchmarking this, and for instance, the in differential abundance testing, the relative transformation seems to work the best. Commonly, the CLR, the centered log ratio transformation, is applied to accommodate the compositional nature of the data, but it seems that at least in differential abundance testing, it's not needed. But there is in the online book there is much more on this topic also.
Thank you, Tuomas. Do you have any other questions? I don't see any other questions on the Framer pad. Does anyone want to unmute and ask directly? We have 1 minute before we can close this. As you ponder on that, Pascal is asking if the recordings for the session will be shared. Yes, I will upload the session on YouTube tonight. You'll be able to follow through if you want to tomorrow morning. Okay. If there are no other questions, I will encourage you to stay connected to our community. We have a community on Zulip as well as a newsletter that's on line 126 and 128 on the Framer pad. Thank you so much for joining this session. Tuomas, thank you so much for guiding us through microbiome data analysis. I really appreciate you taking your time and bringing your expertise to this community that we are growing here in Africa. Thank you everyone for joining. And to close off, I'll just as usual, hand over to Maria to say a few words about how we are able to have this capacity building sessions with the Bioconductor Africa community. Welcome, Maria.
Yeah, thanks, Laura. And thanks again, Tuomas. That was a really clear walkthrough today. And it was very helpful to see how the workflow fits together. And I think sets us up nicely for the statistical analysis next week. And yeah, we hope you're enjoying these seminars for Bioconductor Africa community. And yeah, we look forward to seeing you this time next week for part 3. Thanks, everyone. Bye.
Thank you. Bye.